Hi everyone, I hope you all are fine. Today I will be presenting my project on competitive fitness of Curtobacterium flaccumfaciens, Pathova flaccumfaciens lineages. Let's start with introduction. So as we all know that global food security is in challenge because of the exponential population growth, climate change and all the other issues. And agriculture has a major role to increase the crop productivity and reduce crop yield reduce crop losses so that we can feed the future. But there is one culprit, that is crop diseases. Crop disease is responsible for 20 to 40%, up to 20 to 40% of worldwide crop yield losses. So minimizing those crop diseases will be crucial to meet the future food demand. On all of those diseases, transport is one of the major diseases of mung bean. It is caused by Curtobacterium flaccumfaciens, Pathova flaccumfaciens. And if we are talking about mung bean, it's one of the important pulse crop in Australia, and the growing region are in central and southern Queensland and in northern part of NSW as well. It is important crop for domestic and export market as well. It can be used in various purposes. The symptom of this disease is irregular peppery tan brown interveinal necrosis with chlorotic margin. So it has like irregular patches which has necrotic and chlorotic regions, and it turns into tan brown later on. So if we go a little bit in detail about CFF, it has like genetic diversity, and genetic diversity within CFF poses a significant risk in mung bean. And competitive fitness, studying about competitive fitness of CFF isolates will help providing insight for the disease management strategies, breeding strategies like that. And in this project, we assume that different CFF strains may have different pathogenicity. So, a little bit of background information. As we all know that competitive fitness in bacteria refers to their ability to outcompete others. And we only saw one competitive fitness study where they inoculated 3 bacterial isolates in zebrafish, and they studied competition as pure strain and when co-inoculated. And for this study, we got data, whole genome data linked to transport from Bigafi ETAL. It was the published data from USQ as well, and this project will also use unpublished data from CSS, which is Center for Crop Health at University of Southern Queensland. So let's talk about the aim of this project. So the major aim was to optimize 3 strain-specific PCR assays for various isolates of CFF. As we were going to study competitive fitness between 3 isolates of CFF, it was necessary to optimize the PCR assay for them, and then to inoculate mung bean with mixture of each strain and equivalent concentration of pure strain inoculum, which means we inoculated mung bean plants with individual isolates, and by combining the different concentration and different type of isolates. After that, our aim was to compare disease expression across treatment. The different plants were treated with different kind of mixes and concentrations, so we wanted to compare them. And after doing that, our final aim was to confirm the strain growth dynamic through qPCR when inoculated as pure or in mixer. So if we talk about scope and limitation of this study, the study will of course help to develop effective disease management strategies by developing a comprehensive understanding of CFF's competitive fitness and interaction, because there is no background information about competitive fitness of CFF in mung bean. So this study will be novel, and we are using qPCR for the first time to study competitive fitness of um, plant pathogen CFF. So this is very novel, and we will focus on multiple strains from different lineages. There are like different genetic clusters within CFF, and we're using isolates from each cluster. So the study will be very helpful in future as well, and limited previous studies on CFF pathogen mung bean and like control environment. So these are the limitations, and we didn't have a lot of time, and we were using only 2 cultivars. So these are also the limitations of this study. Let's dive into the originality and motivation. So as I already talked, the limited knowledge exists on competitive fitness of CFF was one of the motivation to study further about this pathogen and isolates within this pathogen. And as I am the frequent consumer of this mung bean, I love mung bean dal. So I was driven to investigate and address this problem. I don't want to be without eating mung bean. That's why I'm also motivated to do this research. And this study will also fill the gap about in understanding competitive fitness of CFF strains and effect of transport in mung bean as well. So these are some of the originality and motivation, and of course we're quantifying CFF using qPCR for the first time. Um, let's talk about some of the literatures that had information about mung bean, CFF, and competition, isolates competition. As we are talking about mung bean, it was originated in Indian subcontinent, and it is member of Vigna genus and subgenus Ceratotropis. It is annual crop. It has height of 60 to 76 centimeter, and it has cylindrical pod with small spherical seeds. And as it is a as it is pulse crop, it has like rhizobium. It has like symbiosis with rhizobium, and it helps to increase nitrogen in the soil and increase soil fertility. And it is great source of protein and used in varieties of food, as a whole grain, as dal, as sprout, as alternative of meat in these days as well. And it is grown at least in 7.3 million hectare worldwide. And in Australia, the production of mung bean began in 1930s, and now it produces around 30,000 to 60,000 tons annually. And some of the popular variety that we use in Australia are Jade AU, Crystal, Onyx AU, Opal AU, Satin second, Celera second, etc. So let's understand about Curtobacterium. So the word Curtobacterium was derived from Curtobacterium, and where curtus means shortened and bacterium means bacteria, and in flaccumfaciens, flaccus means flabby and faciens means creating. So as a whole, it is a bacteria that is short, rounded, and it causes like um, causes wilting in plant. So and it causes around 25% yield loss in mung bean, and it is grown between 27 to 32 degree, which is the best growing temperature for CFF, and it likes hot and humid condition as well. And talking about colonization, it colonizes systematically as a vascular pathogen and survive in plant debris as well, and spread through contaminated seeds, physical opening, and wounds. So as it is contaminating seeds and it spread through seed, it is very hard to control as well. So let's dive into disease symptoms and management. So as we can see here as well, it has scrotches appearance with intravenous necrosis and clear chlorotic margin. So outside of this necrosis region, there is like yellowish chlorotic region, and when this necrosis happen, after that it turns into a tan powder color, so it is called as tan spot disease. So leaves develop ragged appearance, turn into tan and crumble. The best way to prevent CFF from entering the plant is to use healthy seed. As I already talked that contaminated seed, infected seed can spread disease easily. Using healthy seed is the best way to manage CFF. As the study shows that there are no chemical, heat treatment, biological approaches, or cultural strategies that are proven effective in this for this bacteria. So using healthy seed is the best way. And for technique to assess competitive fitness, there are like two techniques: population genetics and direct competition assess. In population genetics, we calculate the rate at which the various pathogens are growing in various environments. On the other hand, direct competition assays utilized the idea of relative fitness of co-inoculated pathogen, and for this assay, we can use molecular marker to track back the pathogen after inoculation. So molecular marker like SNPs can be used, which will help to track back the pathogen after inoculating, and we can compare how they are different, or how what result they gave. So now let's talk about the materials and method that we used in this study. Three isolates of CFF: BRIB 70614, BRIB 623, 70623, and 70610. Three isolates from three genetic cluster of CFF were used. We also used two cultivars of mung bean. One was moderately susceptible, which was ZAU, and another is Opal AU, which was susceptible to tan spot. For this project, we used 17 treatments, 4 replicates, and 4 experiments in randomized plot design. And for the planting, we used Scott Osmocott planting mix, potting mix, and in each pot we used only one seed to grow the plant. So in inoculation, for the inoculation process, we kept isolate at -20 degrees in the freezer before, after growing them into nutrient agar. After that, the colonies were placed into nutrient broth and incubated for 2 days. After 2 days, after the colonies were fully grown in nutrient broth, we centrifuged the broth and resuspended them, diluted with a various concentration to make the concentration we needed. After that, we inoculated the plant using a pipette, as you can see in the video as well. We took 10 microliter of inoculum solution and placed into the stem of the plant just below the trifoliate, and then we used acupuncture needle to inject it inside the plant. After the inoculation, after 10 days of inoculations, we After 10 days of inoculation, we collected data based on visual rating, and also collected plant leaf sample and stem samples. After that, leaf sample and stem samples were dried using freeze dryer and oven dryer. So leaf samples were dried using freeze dryer and oven steam in oven. After that, dry weight of samples were taken, and the dried leaf, we used that to extract DNA by grinding them and transferring them into 2 ml tubes. And with leaf sample, we also inoculated DNA from inoculum sample, which was stored in the fridge. After that, after successfully extracting DNA, we used qPCR to quantify that. Before quantifying them, we had to design primers for each isolates. Primer and probe were designed using primer 3 input tool. After that, for all of them, for all 3 isolates, we did temperature gradient test, which suggested that 60-degree would be best for all of them to detect all of them in triplex assay. And then we tried triplex assay, and it worked successfully, which was good for us. Otherwise, we had to do a lot of tests for each isolates. And after successfully testing triplex assay, we created a standard curve using 10-fold dilution series of mixture of 3 isolates, and we used 2.5 nanogram initial concentration of bacterial DNA to create the standard curve. And after that, we used plant and inoculum sample to quantify DNA, and we compared. So the data were analyzed using Microsoft Excel, RStudio, and CFX Maestro. The first thing we looked at is the mean disease rating in percentage for different treatments. For these 17 treatments, the disease rating was different, and this standard curve, this bar shows the error, standard error, and the latest in here, if there is like no common letter between this bar, it means these treatments are significantly different in terms of affecting it. And as you can see, 23 at 0.5 concentration had the highest disease rating, whereas 14 plus 10 at 0.6 OD concentration had lowest, and the control plant had no disease, which was good. And when we looked at the effect of treatments on total dry weight, 0.1 1 OD concentration had the lowest dry weight, and 0.3 had greatest dry weight, which means 1 OD affected the weight of plant greatly than 0.3, whereas in the disease rating, 0.3 treatments with 0.3 concentration had greater disease expression. And before doing qPCR, we created a standard curve, and these cross marks are for negative and no template control, and the round one were detected and the concentration were identified using standard curve. And this is the comparison of isolate quantities. So the left one is for pure strain inoculum, where the concentration of, the quantity of all the treatments increased after inoculating into the plants. And on the left one, it's for the comparison of isolates in mixer, where 70623 isolates grew more than other, and it also decreased the proportion of other isolates in mixture. As you can see that it starts with 0 to 74.28, and again it starts with 74.28 to 0. It is a little bit hard to understand this curve, but the 70614 and 70610 isolates normally decreased after inoculation, and all the 70623 isolates increased in mixture, which means 70614 outgrew other isolates. And for the future prospect, we can consider conducting biochemical assay to investigate if 70614 actually produce any inhibitor chemicals that compete with other isolates. We can explore the possibility of future studies for like in vitro setting. In vitro setting, we can look at the competition in plates, in like tubes, and we can also extend the duration of plant cultivation. We inoculated them after 14 days of planting and harvested them after 10 days of um, inoculation. So in future we can increase that time so that we can see the effect of that transport in yield as well, and we can also do this experiment in open field as well. So, and we can use multiple cultivars as well. So these are some future prospects, and in conclusion, we had the problem that different genetic diversity within the CFF poses risk in mung bean, and as mung bean is an important crop in Australia and especially in Queensland, we need to do something to control that, and we aim to explore virulence of different strains of CFF using competitive fitness study. For that, we optimized qPCR, inoculated them different isolates at different concentrations on mung bean plants, and we compared disease expressions, confirmed the strain growth dynamic as well, and in the result we found that the quantified isolates, the major success for us was we were able to quantify them using qPCR in a triplet assay, and after quantifying that, we found that 70614 outcompeted other strains and increased its pathogenicity. And looking at the effect of population density, we found that 0.3 OD had more disease expression, and 0.1 OD had more effect on stem weight. And this study, the information from this study can be used in future breeding programs or pathogen control strategies, or in future research that will look at CFF or transport in mung bean. So these are some of the summary of my project, and these are my references. I would like to acknowledge University of Southern Queensland Broadcare Cropping Initiative, BACI, my principal supervisor Noel Knight, my co-supervisor Ahmed Saad Basa Saud, my course examiner Jing Hao Tao, and my friends as well who helped me throughout this process. Thank you so much, everyone. If you have any question, please post it to the forum. I would love to get back to you. Thank you so much.